Welcome to the DNA of Things, where we explore the fascinating intersection of DNA, environment, culture, how this comes together synergistically in our everyday lives. Now, today we have a visit with Dr. Melissa Smith, a leading expert in genomics, personalized medicine, and immune response, recently promoted to associate professor at the University of Louisville and the director of the Sequencing Technology Center there. In our conversation, Dr. Smith shares her journey from studying microbiology and history at UC San Diego through a transformative PhD at Harvard to her groundworking break in industry, specifically in a field that she pioneered, that is immunogenomics. It's very synergistic, to say the least, when complementary fields come together to form a different perspective of, in this case, our biology and huge consequences potentially on the future of precision medicine, and in particular, the development of vaccines and, well, all therapeutics in general. So in this episode, you'll learn about the complexities of antibody diversity, the impact of genetic variation on health and disease, and the exciting future of precision medicine. Our discussion is packed with insights that could reshape your understanding of human health and the importance of inclusive research, that is, the human metagenome. So as we dive into this episode, I will warn you, this is more on the technical side of things. But for those non-technical folks listening, there is a wealth of knowledge for you to ascertain here. And for those more technically inclined and interested in genomics, I think you're going to be fascinated at this intersection of immunology and genomics, that is immunogenomics. At the end of the day, what you're going to appreciate, what you're going to want to come to appreciate, is that there is much more diversity in the single individual than we ever thought before. And especially insofar as our capacity to create antibodies to protect and sustain the life of each individual and our species. So let's dive in. Dr. Melissa Smith, welcome to the DNA of Things.
Thank you so much for having me. I'm so excited to talk to you today.
Oh, well, the feeling is mutual. Your expert background in genomics, personalized medicine, the contributions that you made to understanding host pathogen interactions, immune responses, cancer genomics, really you are at the forefront of some of the most important things, as it relates to human health, wellness, human performance, precision medicine. And here you are an assistant professor at the University of Louisville. And I didn't know this, but you also direct the Sequencing Technology Center. Is that correct?
I do. And actually, I have sort of a fun update that's fun to celebrate with you. So about 10 days ago, I got promoted. So I'm now an associate professor. How much ever that means, but it's tenure, which is a huge milestone for an academic. So very excited about that.
Well, that's massive. Congratulations. That's
fun to say it. I haven't said it much, so it's great to say it out loud.
Wow. Amazing. No small feat, because that's as the way I understand it, a very holistic process. It's your teachabilities, your contribution in grooming graduate students. As you told me, you were actually recently helping your first PhD student enter into the world. There's your publication record. There's the social contributions within the department, across campus. So it really is an all-encompassing endeavor. So yeah, congratulations again.
Thank you.
So we are going to dive into your research, and before we do so, again, this is focused genomics, personalized medicine. We've had some very interesting preliminary conversations on host pathogen interactions. I'm really interested for you to share your knowledge around the, let's say, the adaptive immune response. I think that's where you're expert, and what we're learning there in terms of the call it, interpersonal heterogeneity. So some
Yes,
fascinating things you told me last time. But before
absolutely.
we dive into that, I mentioned to you, this is the hero's journey and you're the hero. So I'd love to hear, your personal journey. What led you into the field of genomics? Were there any key moments that shaped your career and, specifically the academic pursuits that you find yourself faced with today?
Yeah, no, actually, I enjoy talking about my journey, especially to trainees, because it's not linear. And I think very few of our journeys are linear. So it was linear at the beginning. I did my bachelor's work at the University of California, San Diego. Actually, I have a dual degree in microbiology. I was pre-med in the beginning, and then I shifted over to research with some time and exposure. I also have a degree in history, which has actually, I think, been really helpful for my writing skills as I've gone through science, not something that is really focused on in science education, but is so important.
Mhm.
I did my PhD at Harvard University in virology. So actually quite different than genomics. I mean, I did my PhD in the era of Sanger sequencing, so I did plenty of that, but it was not anything like it was today. I studied the interactions between HIV envelope glycoprotein and neutralizing antibodies. So through that work, got very interested in immunology, decided after a 7-year postdoc or PhD, which is, a significant amount of time, that I wanted to sort of make science serve my life a little bit. And I did a postdoc abroad. So I actually did a postdoc at the Institut Pasteur in Paris. And I did it in immunology because I had learned about the virus during my PhD and I wanted to learn more about how the immune system worked. And what I found really fascinating, and I still do, is, the virus and the immune system are constantly interacting inside of an infected individual. But virologists and immunologists rarely talk to each other. And so a lot of the work that is done is in isolation, where the immunologists use the virus as a model, or virologists use antibodies as a model. But really, the two systems interact and co-evolve together in an infected individual, but the fields don't overlap very much. And so I really wanted to learn each so I could find where they did overlap, because that's where I really like to play. After I finished that postdoc in Paris, I did a second postdoc so I could get home. When I came home to San Diego, I did a postdoc at the Center for AIDS Research at the University of California, San Diego. And that's where I started learning genomics. So in that laboratory with Davey Smith and Doug Richmond, we were really interested in understanding the evolution of neutralizing antibodies in HIV infected individuals. And so what we decided to do was to use a really new sequencing technology, single molecule real-time sequencing, or SMART sequencing, from Pacific Biosciences. So this was a sequencing technology that could sequence big pieces of DNA. And in 2012, which is when this happened, a big piece was 3 kb, and which is about the size of the HIV envelope. So we developed a method to sequence the entirety of the envelope as a single molecule. And in doing that in patients over time, we could evaluate the acquisition of mutations and changes suggestive of viral escape from neutralizing antibodies. And pairing that with antibody studies in the serum of those individuals, we could try to understand a bit more about the sort of evolutionary arms race between viruses and the antibody system. Like many people at that stage, I was really burnt out in academic labs. I submitted a ton of postdoctoral fellowships that didn't get funded. And I started to work really closely with my colleagues at Pacific Biosciences because we were really pushing the edge of what that technology could do at the time. And so I needed a lot of help. And so I was reaching out to them a lot. And eventually, an opportunity at PacBio came for someone to move over into the applications development team. And since that was really what I was doing at my postdoc, was developing a novel application for that technology, I jumped at the chance. I think especially trainees consider industry to be a little bit of a magic box. I would say that, and what I say to the trainees that I talk to is that, you don't work any less in industry, that's for sure. But you have a target, right? And so I worked at PacBio for 2 years. So not very long on the calendar, but I would say that it was probably the most formative experience in my career I've ever had. So it taught me about being on a team. So instead of a single investigator competing against others in the field, we were all on the same team, hundreds of us, aiming for the same goal. It taught me about pivoting and being agile to sort of pick up any job that was needed. I came in to work in the laboratory, but I ended up working in marketing because obviously I like to talk about
Hmm.
the work that I do. And so, it gave me a lot of experience and more than anything, a lot of confidence, and sort of showed me that my skill sets were valuable. I've been surrounded by people who have the same knowledge as me for a long time. So to be in an environment where the knowledge set that I had was really unique made me realize that I have something to offer in sort of the wider space. And so through that practical experience, I started to learn about genomics. So of course, the first thing I learned about was PacBio, because that's where I was working. After about 2 years of doing this, I traveled on the road a lot. I went out with the sales team and met investigators that were considering buying the instrument, heard what science they were doing. I was getting more and more excited about the science that was capable of being done using this instrument, and less excited about the marketing development of flyers and brochures and going to conferences. And so I looked for an opportunity to sort of make a leap. And I was incredibly fortunate to work with Bobby Sebra. So he also used to work at PacBio, but at this point he had moved over to the Icahn School of Medicine at Mount Sinai in New York City, and he was running their Genomic Technology Center. He and I actually had a very similar background in terms of what we were doing at PacBio and development of new techniques. And so he thought, my team is growing here at Sinai. I would love an assistant director to come in to help manage day-to-day operations. And it gave me a sort of a back door back into academics. So I came in at Mount Sinai as the assistant director of genomics technology and also as an assistant professor, because the faculty titles sort of went along with it. So that was, incredibly lucky. I have to say, as an aside, and I think it's always interesting for trainees in particular, I don't think I've ever cold emailed for a job. I think all of the jobs that I've gotten throughout my career have been because of conversations, likely over a beer, about frustrations or excitement or things that I'm interested in that may not be working with the jobs that I have currently. And so, keeping that network alive and accessing that network when you need it, is one of the biggest things I've learned throughout my career. So I worked with Bobby for 4 years. In the time that we worked together, our team grew from about 12 to 35, and we managed a massive genomics core. So that was my boots on the ground learning about the rest of genomics technology. And we were so lucky at Mount Sinai. They have incredible support for their genomics endeavors there. We had sort of every instrument you can imagine under our roof. So we had Illumina, PacBio, Oxford Nanopore, 10x Genomics. We had the Berkeley Lights, like cell on a chip system, which is amazing. So it really gave me an exposure to sort of everything that you could wish for and what you could do with it. And Bobby himself is an incredibly supportive individual. You asked me before who I think would be a great person to have on this podcast. He would be at the top of my list for the work that he does sort of across the realm. And he supported me and gave me sort of a small fund to develop my own projects in the lab in addition to managing the core. And so I worked with someone I had identified through the literature who was working in immunogenomics. So immunogenomics is just sort of the organization of the DNA that encode our antibody and T cell receptors. It means a lot of things, but that's how I think of it. And Corey Watson, who I found in the literature, he had developed and published several reference sequences for this part of the genome. He had actually been at Mount Sinai, but had since moved to University of Louisville. And so we started working together remotely. We wrote a grant together to do some population, so to develop a method to sequence these regions of the genome. They're very highly repetitive, not easily resolved with other technologies. So long reads were really essential. And just trying to understand how diverse they were in populations. They're antibodies. They're meant to detect anything that you can possibly be exposed to, but there's a lot of misunderstanding or misrepresentation in the literature about whether they're different between individuals. And so we started on this endeavor in 2017, I guess 2015, but our first grant was funded in 2017, and we never really looked back. And so when COVID hit in New York City in 2020, our team never sheltered in place, never stayed home. Our team was in the laboratory every day. We were sequencing every isolate of SARS-CoV-2 that came through the Mount Sinai Hospital. We're actually on the science paper of the molecular evidence of SARS-CoV-2 introduction in New York City. We sequenced those first isolates on the East Coast. And it was shocking to be in New York during that time. I had a small child. As a virologist, if you would ask me 10 years ago what I would want to be a part of in my career, I would have said some type of global pathogen outbreak. I would have thought that discovering a new virus was super cool, being a virologist. But now that I'm a bit older and I'm a parent and I was doing a 3-hour commute in and out of New York every day, I thought that maybe it was time for a change. And so I reached out to Corey because we work so closely together here and asked if there would be the possibility of me coming to Louisville. And through a lot of work, the administration here, a lot of selling of that ability to profile SARS genomically, because at the time that was the highest priority for everyone around the country. I was able to move in July of 2020. So I'm here now. It's just finished my 4th anniversary. it's a great place to be. I think, people don't think of University of Louisville when they think of great genomics, and I think that's a mistake. I think we should. I think you can do good science anywhere as long as you have the facilities and the collaborators. And we have a great team here. I'm so proud to be part of this department here at Louisville.
I think I told you in our last conversation that I had a brief stopover in Louisville, Kentucky, just on some entrepreneurial work that I was doing, and I was very disappointed to not be able to find the original Kentucky Fried Chicken.
Yeah, yeah.
But to your point, having me myself been thinking about DNA for 25 years, Louisville doesn't come to the top of mind. But your journey and the kind of research that you're doing, just couldn't be more cutting edge. And I wanted to kind of peel back some of the layers because, the great thing about, our audience is you've got super technical people, you've got super entrepreneurial biz dev type people, you've got, folks who are, looking to unlock human potential on the day to day. And I think you're a very special guest because, as evidenced by your journey, you've traversed those different domains, especially, enlightening to hear not only like the microbiology background and then the kind of tentative dance with the medical system, but then going around it into graduate school. I was faced with a similar fork in the road, and I just couldn't, the idea of 2 more years at a desk was just not appealing to me. And I love the research aspect of things. And as you described it, being very different from industry, you're really, depending on your supervisor, you're trailblazing, right? And so that's not for the faint of heart, to be sure. But it's fascinating your background in history as well, like liberal arts, and it makes me think of somebody like Stephen Jay Gould, who I didn't know this, but I only learned it recently. Obviously, he had written to the National Natural History magazine for I think it was like 20 years or maybe nonstop, and obviously wonderful writer. But he was actually one of the editors of the Great Books series that J. Adler Mortimer put together, which
that's awesome.
Wasn't a surprise, but he was, and it sounds like you are, just an incredible, speaker. And, that's a rare find in terms of having somebody who deeply understands the technology, but has an art for the knowledge translation, and then add that with the fact that you've been in industry, whereas I think, one of the things you talked about, like from an entrepreneurial perspective, how to be agile, how to fill in, see a need, fill a need. But then there's also like the accountability. One of the things I saw in academia, just from experience, is there really isn't much accountability. Is once you get your grant, it's, if you publish, then you'll probably get more grants. But then it's like, well, did your publication actually make it into the mainstream? Did it actually make a difference? If so, how do we know that? So it's as I say, a rare find to speak with somebody who, has that just robust, holistic field of experience. Now, in terms of where we are in your current research path, you were talking to a certain extent about the interpersonal variation of, would you say, antibody production as encoded down to the level of the DNA, and I suppose the expression profiles as well. Could you maybe just drill in, to that at like maybe an undergraduate level, but also like at a level like, I don't know, maybe a 10 or 12-year-old might understand in terms of like, how do we make antibodies, if you could break down into like maybe a simple 3-step process. And then, how do vaccines work, for example? And then how might your insight into this genomic detail provide new ways of thinking about let's call it vaccination in the realm of precision medicine? So I want to make sure I'm not thinking about it the wrong way, because I kind of see it as likens to when in the world of the development of therapeutics, you've got to account, if you want a drug to have efficacy broadly, then you need to account for the human metagenome, not just
absolutely
right. And so it sounds like you drill into how our immune system works. And again, I want to just shine a light on something else you said that's super important, is immunologists and people who study genomics aren't necessarily talking. And then here you are, there's this interdisciplinary synergy that's happening. So yeah, just
Yes.
broadly speaking, how do we think about immunology? What
Sure.
are you telling us about the diversity of genomics? What does that mean for precision medicine, for the development of vaccines, and then maybe precision medicine more broadly, autoimmune diseases, et cetera? Mm-hmm.
Yeah, sure. So I'll start at the beginning and I'll go through the process. I thought your sort of three-step request was really a good way to structure it. So unlike most genes in our genome, where we have one copy from our mom and one copy from our dad, we express either both copies or one or the other, depending on structure and regulation. The antibody and T cells, so we do both arms of the immune system, is is structured in the genome such that there can be an incredible amount of combinatorial diversity. So what do I mean by that? I mean the antibody itself. So the antibody I'm going to use as the model, but the same mechanism occurs for T cells. So the molecule itself is shaped by like a Y, and the arms of the Y are what I call the business end. So this is the end of the molecule that interacts with a virus or a bacteria or a piece of tumor cell. And in order to be sort of ready for any possible pathogen or non-self that it can encounter, it has to have an incredible amount of diversity, right? And your body, when you're born or conceived, even you don't know what a flu particle looks like or a measles particle, right? So what it does at the DNA level is that there's arrays of genes that are very similar. So there's a gene family called the V genes, and there's over 100 of those. There's D genes, diversity genes, and there's joining genes, J genes, and they undergo a somatic recombination. So every B cell, every B cell as part of its development will choose one V gene, one D gene, and one J gene and recombine them into a single cassette. And that is the region at the tip of the Y that encounters the antigen. When they ligate together, when they glue together into their cassette, so they delete out any of the DNA in between and it gets discarded. And when they glue together, it's not just a straight DNA repair or a ligase. There's an addition of non-templated nucleotides. So non-templated bases, a small string of A's, C's, T's, or G's, but in a random order that's not encoded by the genome that is used as that glue. And so not only do you have the combinatorial diversity of any of these 100, any of these 27, any of these 9, but then you also have the addition of these strings of bases that can encode specific codons in between each of those cassette pieces. So together, when you think about what that diversity, and then so that's the heavy chain, that's the stem of the Y with the first arm, and then there's a little arm underneath called the light chain. It does a very similar process. It has a V and a J. There's no D. So also combinatorial diverse stuck together with extra bases. And then there's both the diversity of picking one of the V's, one of the D's, one of the J's, and whatever the glue is in between. Then there's the diversity of that and the light chain and how they mix and match. And together, that combinatorial diversity gives you about 1 trillion, 1 times 10, 1 billion, 1 times 10 to the ninth possible combinations. And then that's expressed on a B cell. It's called a naive B cell. It's a B cell that hasn't encountered whatever that receptor is now specific for. And that hangs out in lymph nodes. It swims around in your circulation. When it encounters something that it can bind to, so it's slightly specific for it and it can attach, then it undergoes further maturation and it acquires additional mutations to try to enhance that attachment. And that's called affinity maturation. And when you add that additional mutational process to try to enhance the affinity and enhance the avidity, so that the tightness of the binding and the specificity of the binding to that recognized particle, then you get up to sort of 1 in 10 to the 13th possible combinations. So really I heard someone today on we had a journal club this morning and they said, just to put that in perspective, that's more than the trees on the earth and that's more than the fish in the sea. So just the number is almost too much. Actually, it is too much for me to comprehend. But that variation, is out there to be ready, to be primed. So when you're vaccinated, what happens is that you receive a virus or a piece of a virus. We'll just take a viral vaccine as an example, even though there are lots of bacterial vaccines out there as well. You receive a virus. In the case of measles, it's a whole live virus. In the case of like hepatitis B, it's a piece of the virus. that will bind those antibodies, get that maturation process started, and make a group of B cells that are ready to fight that virus if they see it again. But because it's only one dose or a few doses, those B cells will then calm down over time, but they'll stay. They'll circulate throughout your lifetime, or at least for a long period of time until you get boosted again. And they'll be ready and what we call primed to react if they ever see it again. Because the next time a specific antibody or B cell sees that antigen, it doesn't have to go through all of those maturation steps. It doesn't have to go through all of those mutational steps. It can just respond, and it's highly specific and tightly binding, and it can just initiate a very well-coordinated immune response. So the only difference between that and T cells is that T cells don't undergo that further maturation, that mutational process called affinity maturation, but the same combinatorial diversity is there. And so you have, for T cells 10 to the 9th, for B cells 10 to the 13th, just an extreme amount of diversity. So if you look at a general immunology textbook, our favorite is by Janeway, and you look into this process, and I want to apologize ahead of time. I sort of always make this joke when I give a talk. Anyone who's learned VDJ recombination in class, it's almost like a PTSD reaction. It's so complicated. It gets really overwhelming, and you can sort of see people cringe when you bring it up in a presentation, but it is really important. We learn in the textbooks that we all have the same V, D, and J genes that can go into this recombination process. And that really, it's a random reassortment. And that random reassortment means that you and I will have inherently different combinations just because my genome recombined differently than your genome. Right. So what we set out to do in that first grant in 2017 was to develop a method where we could sequence just that locus of the genome because it wasn't resolved in the original Human Genome Project. It was too complicated. These little cassettes are very small. They're a couple of hundred base pairs, and they're highly similar and highly repetitive. And so when you use short read sequencing like Sanger or next generation sequencing methods, you can't really assemble. They all look alike. They mismap. You can't make a contiguous picture of the locus. But if you use these long reads, right, that are 10 kb, I think of it like a jigsaw puzzle. Yep. Like if you have a sunset as a jigsaw puzzle and it's 10,000 pieces, you're going to have that one like purplish piece that you're never going to know where it goes. But if that picture is 100 pieces instead, you have a lot more context. There's a cloud here or a piece of the beach here. It's much easier to know where it goes, and you can much more easily create a complete picture. And so that's how we use long reads. We use long reads as big puzzle pieces. So instead of capturing one V gene at a time with a short read, we were capturing 10. Right. And that allowed it to have enough context and enough variation that we could then accurately stitch it together. The whole locus is about a megabase for the heavy chain variable region genes. And so we did about 180 individuals in that first grant from 2017 to 2023 with this targeted assay. And we found more variation than you would even know what to do. Like we knew we'd find variation, right? But we didn't realize how extreme it would be. So not only do you have, so the first level of variation is just structural. So if you have these sort of collective, say 140 V, D, and J genes, let's just pick that number. Some people will have just deletions in their genome where they're missing 10 genes. And these people are walking around seemingly healthy. But if you were to have some type of drug that targeted that particular gene, that person would not be able to respond as planned. Now, they may have a different response, but as planned, it wouldn't go as well as thought. So what we found, we found these big deletions. We found individuals that had 25% of their locus deleted, which was crazy. But we also found that at the per gene level, there's not, and so these genes come in families. There's a V1 family and a V2 family, and then within these families are specific genes, so say V1-2. But even within that specific V1-2, my V1-2 may not be the same as your V1-2. There are different flavors. There's up to 10 in V1-2, because that's a real gene. And we call those alleles. So it's a slightly different use of the same words that we used for some autosomal genes, but because it's such a complex structure, it's slightly off. These alleles are just different flavors of genes that are defined by nucleotide changes. So sometimes they're coding changes, sometimes they're non-coding changes. In the case of V1-2, there's single residue changes that completely change the function of that gene. And so we looked across a lot of global populations, big, like sort of high level global populations, ancestral populations and these 180 individuals and just said, wait, so much more variation than we thought. But like, does it actually matter? So to your point, we also looked at the RNA level at which antibodies were being expressed. And then we did a quantitative trait locus analysis, loci analysis. So looking if a variant in the DNA impacts the ability of a gene to be expressed at the RNA level. And we saw that absolutely, yes, that's the case. In fact, 70% of the antibody genes were impacted by some kind of variation.
Wow.
In the DNA. So it was really a wake-up call for us that we knew there was something there, but we didn't know how big it was. And we also didn't know how it would trend with populations. So people within ancestrally related populations are more similar than across ancestral populations. And this was our first like big light bulb moment of, you know, these are the genes that are being targeted during infection, during immunotherapy, during vaccination, where knowing this context could be really important. And it's something no one's ever thought of before because the paradigm was that we were all the same and we just had random mixing and matching that was different. So we published this last year in Nature Communications. And it really, I believe, is a hugely paradigm shifting moment for the field. We've done the same in T cells and see very similar levels of variation there. A little less complicated, but still quite complicated. Again, something that's not been considered. And I think when you talk about drug efficacy, whether that's a vaccine or another immune-related drug, most of the discussion about when things work and when things don't work is about the formulation itself, right? Do we have the right piece of the virus? Are we using the right adjuvant?
Right.
I think we also need to start incorporating, does the host respond as expected? Do they have the right sort of immunogenomic context to actually be able to respond? Because otherwise we're chasing down a road that's never going to work, right? Because the DNA is the foundation of everything.
So you mentioned the dichotomy of disciplines, genomics and immunologists. So I'm not schooled in immunology, so I wanted to see how much of this I actually followed and maybe where my brain took the wrong turn. So I think I understand, the call it hypervariable regions of the genome that you're talking about quantifying, having been previously near impossible to capture a sequence on because of the nature of, say, next generation sequencing, which tends to kind of truncate the length of DNA to be very, very small, much smaller than, what were you saying, like 2 kb, or how long were these strands that were
we're sequencing 10 kb strands, but next gen sequencing is really limited to up to like 500 base pairs. So,
exactly, for 0.5 kb. So in the technology depends on basically bioinformatic algorithms to align these sequences and form these contiguous arrays. So it's kind of like you bash apart a piece of the genome in a giant puzzle, and you can look at the box and kind of line up the outside of the puzzle, and But I think what you're saying is these regions, these hypervariable regions of the genome, are like all these kind of like puzzle pieces that have no context, and so you can't really align them.
Absolutely.
So you've created.
That's perfect.
Yeah. So you've created.
And the puzzle piece is much bigger, so they have more context to be able to put them together with more confidence, right? Like it's critically important. Yeah.
Got it. So you're missing a incredibly important aspect of our humanity, of the survival of our humanities, frankly.
Right.
Frankly.
Right.
Let me see if this is also analogous to another system that I studied called more of a bacterial system. I wouldn't call it an immune system, although I could stretch it and say parts of it were. And so what I'm talking about is the integron gene cassette system. You studied bacteriology. Yeah. So basically these are these arrays that can be, I don't know, I think some of them are as long as at least 15 kb, maybe 150 kb, and they include these cassettes, which you're talking about, and they have these repeat areas in the genome that allow for this constant shuffling and recombination.
Yes, absolutely.
And you talked about business end of the molecule, so to speak. Now, these genes they're in bacteria, so they're not humans, but they would code for invasive genes, perhaps to invade a host, or protective genes from microbiological warfare. So we find them in like coral mucus, we find them in our gut microbiota, and so there's all this very similar evolutionary pressures, right? Sort of needing to be ready for a lot of different potential targets. Right. So, and so then the next question was, these cells that you these people that you sequence, you mentioned somatic cells. Which cells were you sequencing? The Bs, the T? Were you isolating those, or how were you getting to the genome sequence? Was it cell
Right.
or cheek cells, or where did it come from?
Right. So we've tried this in a lot of different cells. Actually, we're able to get it out of just a PBMC cell isolation. So even though like the majority of PBMCs are B cells and T cells,
Mhm.
the DNA in the non-B cells is not recombined, and we configure the germline array out from those. And the DNA in the non-T cells includes the T cell receptor not recombined. And so there's enough non-B, non-T for us to be able to get enough DNA to recombine them. So that's great
Yeah.
for us, right? Because it's the most easily accessible cell type from an individual, just a simple blood draw, not invasive.
Got
But
it.
we have been able to do this also from uh, tissue biopsy in animals. So we also do this in rhesus macaque, which is a very popular animal model for infectious disease and vaccination. Really important to also know their antibody and T cell genes. We've done this from cheek swabs. We've done this from tumor. So, I think the main criteria for what we need to be able to do this kind of examination is just reasonable quality DNA. So DNA that's bigger than 10 kb, because of course, if we want to stitch together 10,000 base pair pieces, we can't have pieces that are smaller. But other than that, it's been pretty ubiquitously successful. The
Mm.
quality of the DNA, like anything with sequencing, and you know this, the better the quality going in, the better the data coming out.
Sure. Yeah.
Yeah.
Something else I wanted to point to is, the difference between like genomics and genetics, right? So you think about,
Right.
when a sperm and an egg fuse, and there's this, well, actually, even before that, the production of sperm and egg, the whole meiotic process and recombination that's happening there to create variation in the next generation. And then,
Right.
when sperm and egg fuse, that crossing over, how, what's going on there to again add more diversity of phenotypes, so that, essentially we look to natural selection. We're required that a certain amount of diversity exists and the environment selects. And, very broadly speaking in Darwinian terms. And so if your diversity is very low, you're not going to go on very far as a species. But
No.
the thing that I'm pointing on here is like with genomics, very different than, genetics, or more all encompassing, I would say, because we're talking about
Right.
although your genome is in each cell, there's a dynamic processes that are going on. Listeners have probably heard a lot about, well, epigenetics, for example, the gene
Right.
expression and silencing in different cell types, for example, and based on exposures to different stressors. The fact is that kind of recombination that was happening, say, once at gamete formation and then again at zygote formation, it actually seems to be happening in a very dynamic fashion in these immune system cells. And
Yep.
so that is to be ready for anything, as you say. I wanted to, because I know you specialize also in, I think, autoimmunity to an extent, and we talk about cancer genomics. What is going on when the immune system decides to attack itself, and is your research giving any clues as to, who might be creating a higher population of antibodies with affinity to self, and
Sure.
is there a way to follow that community in an individual's lifetime? Are they, because I'm assuming here it's like more inflammation, more activation of the immune system,
Right.
more likelihood that you have like self autoimmunity, more likely you're going to trigger a flare up. I'm speaking in very general terms, but yeah, to what extent is your research helping us better understand that autoimmunity side of things?
Right. I'm going to get there. I just want to comment on one thing for what you said previously. So, I think epigenetics is incredibly important. It's something that I'm currently still learning about because it wasn't well known and well characterized when I was studying and in school. So I'm sort of having to learn it on the fly. I think one of the neat things, method that we developed is it is a DNA based method. So, and when you think about things that regulate expression, right, it's coding. So if you have a non-synonymous mutation that changes the protein coding is one thing, but a huge amount of variation also it occurs in non-coding regions, right? The regions in between the genes where enhancers bind or promoters are. And so we see an incredible amount of variation in these non-coding regions that they may control if these genes can be expressed because they can't recombine correctly. So we're really excited to chase down a lot of the epigenetic control, a lot of the non-coding regulatory control, again, to try to understand how the genetic structure impacts expression. Now, getting
Yeah.
to autoimmunity, what you stated is exactly what we'd like to do. That's what we're aiming for. So in addition to the genomic assay, we also built an RNA-based assay. This one's called FLAIR-seq. So why I laughed when you said FLAIR a minute ago. So full-length adaptive immune receptor repertoire is sort of a mouthful. But the idea is, again, when you think about that antibody structure, the vast majority of sequencing-based profiling of repertoires really focuses on that variable region, that end, that business end of the molecule that binds the antigen. That's understood. People want to figure out how to use sequence data to predict antigen specificity. But it turns out that the stem of that molecule, the stem of the Y, actually is what mediates a lot of the function. So it's called the FC region, the constant domain. It's what binds to receptors on macrophages to promote engulfment. It binds on NK cells to promote antibody-dependent cellular cytotoxicity. It binds all of the complement proteins to kick off the complement cascade. So it coordinates adaptive and innate immune responses. And I firmly believe that just because of a misnomer of semantics by someone calling it constant, because of course it's constant compared to the crazy variability we just talked about, no one has looked at it. And so we developed this assay that could sequence the entirety of the antibody molecule, the variable region, and the constant region. And we started to look at how much variation is actually in the constant region, and can we predict how that variation would impact function downstream. And where we started was actually with autoimmunity, and a few reasons for that. One, because it's a huge unmet need, right, to understand why these antibodies are starting to target self. A lot of those diseases have a lot of detail already known, that they're specifically antibody mediated, that they're specifically complement mediated. So a lot of the mechanisms already link what we think will be involved in this functional genomics assessment. And so we've been working in the context of myasthenia gravis, which is a autoimmune disease that targets the neuromuscular junction. It blocks acetylcholine receptor, acetylcholine interaction, and inhibits mutations. And I keep pointing at my face because it starts in the face. You get eye drooping, cheek drooping. It can eventually impact your ability to swallow and breathe. So it's a really horrible disease. But people have identified already that it is mediated by the IgG1 type of antibody and the IgG3 type of antibody. And so we were able to profile individuals with the disease using our long read antibody profiling method, FLAIR-seq, to identify the IgG3 specific signatures. And what we see is that there's definitely mutations in that constant region that are known to be associated with like enhanced complement function. They're not in everyone, right? They're in very specific alleles in very specific people. And so what we're doing now to follow up on this, right, is to back engineer these mutations we've seen in the population survey in vitro and confirm that they actually enhance complement activity. So it may not be so much that you have a particular antibody specificity that hits self, that, you know, attacks self. It may be that you have an antibody that everyone else has, but you have a constant region mutation that sort of overacts, right?
Yeah.
So it binds, and instead of just being nonchalant, it recruits complement, starts destroying your receptor. And so what we're really interested in is, obviously we need to confirm this and validate it and show that it's a functional implication. But if that is true, right, if we're able to say that, we know that NG, like most autoimmune diseases, is more of a syndromic diagnosis, and that there are several mechanisms that lead to the same disease. Only one of those mechanisms is complement mediation. But a lot of people are given complement inhibitors just to see if they'll help. Now, if we can say that the people who have complement mediated disease have these specific genetic variants, then we can help target the right therapy to the right people who are going to benefit from it, and say, no, these people aren't going to benefit from it. They have a different mechanism. And then they don't have to deal with the side effects of an immunosuppressive agent, right? And so I think this is where we're getting into that precision medicine, that the potential of our work is really to hopefully identify signatures that are immune related, so B cell or T cell signatures that give us an idea of function or an idea of responsiveness, such that we can get the right medicine, the effective medicines, to the right people, and then avoid side effects from medicines we know aren't going to help because we can tell that their immune systems are not responding to that particular pathway. And I think we've got a lot of hints about that, a little bit in the autoimmunity space, a little bit in the vaccination space. We're doing some work right now in the cancer immunotherapy space. But this is where, you know, another thing, you know, that we're really passionate about, right, is it's about population diversity, but all populations, right? It's a diverse human family of populations. And a lot of these populations, as I'm sure you've discussed on prior podcasts, have been really understudied in genomics research. And so we want to bring them to the fore as well and make sure that we understand their immune system so the understudied populations are getting the right treatments as well.
Mm. I mean, you could imagine a scenario where different populations have lower, call it interpopulation variation, and some have higher. So you're probably going to do some like diversity studies, molecular ecology, and say,
Yep.
okay, yeah, okay.
Exactly.
Yeah.
Yeah. There's a really great example in chimps. And I remember hearing this when I was a graduate student. It was a group in Rotterdam. I can't pull out the name from my head. It's been 20 years, but there's a known HLA molecule. So the human leukocyte antigen that recognizes T cells that are specific for a virus. And there's a known allele of HLA that accelerates HIV disease. It makes you a more rapid progressor. It makes your disease worse. And we know that. We've identified it in the context of HIV. We screen HLA and HIV patients to try to predict sort of how they'll progress. But if you look in the chimpanzee population, there are no chimpanzees with that allele. It's gone in their genome. And the thought that was published by this group who works in Rotterdam at their primate center was that there was a wave of selection that came through when SIV chimpanzee, the SIV affects chimpanzees, swept through the chimp population. Those animals that had that allele were killed before they could reproduce because it advanced their disease so rapidly, right? And the fact that it's not there anymore means that was a bottleneck. And so that's a really great example of how immune genes, for better or for worse, can impact your response to disease and at a very, very high, 100,000 foot level influence evolution.
Let's tap into a little bit of that right brain of yours on the history side of things. And so I'm thinking, with respect to one of the things you said, the intronic regions of the DNA of the genome. So my PhD supervisor was Dr. Ford Doolittle, who just loved the selfish DNA hypothesis. He was big on deconstructing Darwin's tree of life, at least, how it might have been built relative to more of the microbial population. I'm going to go to like the ENCODE project. I don't know if you remember that. Yeah. Okay.
Yep.
And he actually wrote a rebuttal to that, which was, and it was taken up in proceedings of National Academy of Science, where, essentially the ENCODE funding was to be attributed to those who were out to prove function. And then, of course, his philosophical question is, because he was much more of a philosopher. I mean, he did his undergraduate Harvard, PhD Stanford. He worked with the Russian doc that got the White House medal for proving or setting the paradigm of one gene, one protein, one function. I forget his name.
Yep. Yeah.
So, of course, I think just Ford was contrarian by nature. And I think he'd been asking that question, well, what do you mean by function, since his graduate school days? So, because it is nuanced, but it's important, because I think a lot of this, like this notion of junk DNA, it's like, oh, there's, let's say for the sake of argument, 30,000 genes, and that's only accounting for, 30% of the genome roughly. And so what does all this, what does this 70% other stuff do? Well, it doesn't code genes, ergo it must be junk. And, it's just really fascinating to go through this conversation with you today. And with all the tools that are being developed, that you helped develop, we can now actually start to see the dynamic nature, of genomics itself. And so the, function, let's say, of those non-coding regions, it seems to me has a role to play in, tertiary structure.
Absolutely.
The stoichiometry of gene accessibility, gene concentrations.
Yep.
So
recognition motifs for enzymes that enact recombination. I think for us, lots of things came up when you were speaking, but I think pertinent to the conversation we're having right now is that there are methods out there in the immunogenomics space to take an RNA-seq profile of your antibody repertoire and then impute the germline. So based on the genes that are seen in the repertoire, we can impute the germline.
Right.
But what we've seen is that there are non-coding SNPs and non-coding variation that can inhibit the ability to get a gene into the repertoire. So those imputation methods, because they're based in RNA, don't see those non-coding regions and may not accurately represent the germline. And so that's something we're seeing it directly. And I mean, as all of us know, right, we're, trust but verify. And so being able to see it with your own eyes and it is incredibly powerful.
Yeah. I mean, those Yeah, the excision of those intronic regions. Yeah, I always laugh when I go to the grocery store because I think that the parking lot analogy is so beautiful, right? You see, say there's, you're parked and you were fine when you went in the grocery store. There's somebody else parked next to you, and you were, good spacing between all the cars, and then you come back and somebody's like super close to you, and you're like, oh, what a jerk! Why would they do that? And so you're
Yeah.
blaming, but actually it turns out before they parked next to them, there was somebody that forced that initiative over, right? And so you kind of lose that evidence if
Right.
you were to confine your study simply to the RNA-seq molecule and not have the genomic context. And so you would lose that natural history that's
Exactly.
happening in a single body, which is just amazing. So here's something.
In our case, a single cell, right? Because every cell does this differently in the immune system. So in 10 to the 13th cells,
Yeah,
theoretically, right? So
right.
it's pretty amazing.
So do you imagine there's two things, let's say I want to maybe use to springboard us into the future, maybe three things. One of them is, mRNA vaccines, which, by
Right.
the way, when I was in my undergrad, I, studied, HIV, and I thought the promise of mRNA vaccines was like, oh my gosh, you can actually get ahead of it, and you're starting to see, just the incredible speed at which boosters are being made and following
Right.
on. And so that, I mean, it's amazing and scary because it's like overnight the pharmaceutical industry has, and I'm speaking now to your industry mind, right?
Yeah.
The pharmaceutical industry has adopted the Microsoft software as a service model, right? Which
Yeah.
which actually is brilliant because it amortizes the cost of the initial setup and the development, and you're into the long haul, and how are you going to get ahead of everything, and
Right.
so I mean, it's like the nerd technician in me loves that, right? But then there's, so like analogous to this is like, scientists who split the atom. They're like, oh, like it's like there's if we do this, we could harness, energy beyond belief, nuclear energy, or we
Right.
could blow ourselves up. And
Right.
there's just some scientists who are just like, this is so cool, just and so you're not
Yeah.
even,
Right.
so it's like, I think I'm getting into some kind of like a practical pursuit, the way forward, but then also like, morally and considering our humanity, how do we do this responsibly? How do we take account of the potential benefits, but are and intended benefits, but what about the unintended consequences? But in the scope of this frame, this canvas that we have that's emergent, that is, we have whole genome sequencing, we've digitized our genomes, which I think is not enough, by the way, just to get like your
No.
whole genome sequencing based on your cell. It's like, okay, cool, that's part of it. But then there's
Right.
like this whole dynamic thing going on in your body. And then you've got, AI. And so you can imagine much more comprehensive network analysis, much more comprehensive assessment of, the molecular ecology in a single individual. And
Right.
so do you envision new tools coming forward to get a sense of how an individual's their own molecular community of cells are growing through life in response to environmental pressures, behavioral changes, inborn genetic mutation, and so then I want to fit together, mRNA vaccines into that. So it's like
Right.
this the speed at which and the precision at which we're able to, activate our immune systems, is clearly not precise today based on the studies that you're making. So like, what is
Right.
the path forward? And I guess here's what I'm struggling with. Sorry, I had to talk through that to understand my real.
That's okay. I totally understand.
So my real question, I think, is, aren't we? So you think about the rare disease community. It's like, aren't we all rare? And so like
Yeah.
at what point do the big rocks fail? And is
Right.
it going to be cost effective truly to do precision medicine given this new layer of complexity that you're uncovering and
Right.
yet to uncover? So that's my question.
Yeah.
Yeah.
No, I think it's a really great one. And I think I think there's a really important distinction to make between precision and personalized,
Yes. Yes.
right? So I think that from a scalable, cost effective, will anyone ever make a therapy per person? I don't think that that's a reasonable expectation in today's world yet with the technologies that we have.
Yeah.
However, and I don't think that for what we're discovering, I don't necessarily think that's even required. As I mentioned previously, so we took those 180 individuals that we first studied, sort of defined the variation we saw, which was more than we thought. And now we have a project where we're sequencing between 5 and 10,000 individuals
Wow.
from 58 subpopulations, including multiple populations that have never been sequenced before, several indigenous populations around the world. And at least in the first sort of 12 to 1500 that we've done thus far, we're just finishing up, I think, year one of the second phase. That similarity between ancestral population groups is strong, and there is differences obviously between them, but many fewer differences there than like across populations, right? Inter versus intra variation. And so, I think that yes, the big rocks will fall because there is not a one shot fits all. I love the idea of a universal vaccine. I am a virologist. I think that's amazing. I'm not going to say it's not going to work. I'm not going to poke that bear, but I'm going to say that there's a lot more that we need to know about what those vaccines target and how those targets are different across populations to provide more confidence. For me personally, I think that maybe and I think there's two ways to do this, and we can bring your mRNA discussion in, right? There's obviously, almost 30, but more than 30 vaccines out there that all depend on B-cell responses for the most part. So any vaccine that's not a live virus is going to be mainly antibody driven, not live or mRNA. It's going to be mainly antibody driven. For those vaccines that maybe, you see differential efficacy across populations, maybe there's the ability to complement or boost those existing vaccines with a vaccine that's more targeted towards a population that has a different immune profile.
Right.
Maybe we can enhance that product line with boost. And since mRNA is an effective, rapidly developing, straightforward, path compared to some of the other modalities, that would be a good thing to do. I think it's a great way to respond to the information that we may be providing in a specific context. I think moving forward, if I had my dream, we'd be working with drug developers at the preclinical, at the development stage, right?
Mm-hmm.
So say you have 10 candidates for whatever the drug or vaccine is, you test it in a small group of individuals, and we help work with you to identify which one of those candidates has the most breadth of response, which ones would be most active across populations. And here's where I'll de-stealth myself a little bit. You know this, but I think this is a great opportunity to bring this up. Dr. Watson, Corey Watson, and I have been seeing this happening. We've been seeing this evolving for almost 10 years. We really believe in the power of understanding this diversity to provide effective and equitable healthcare around the world. And to do that, we've actually formed a company. So we're in the funding stages. We're very, very early on, but it's fun to come on this podcast and sort of de-stealth. So we formed this company. It's called Clario Biosciences, where we have a website, clariobio.com. But our goal with that company is to really make these assays and these methods and these approaches accessible to anyone, be it academic research, a pharmaceutical drug development group, epidemiology and population geneticists who really want to understand how the diversity in these loci are impacting their scientific questions. And so, as you well know, in an academic lab, we have limitations on staff and funding and scale. And so in order to really make this available and to bring it to the power that it can be, to chase all these paths that we've talked about today, because there's incredible potential of how to use immunogenomic signatures for biomarkers or to guide clinical care, assuming that, the function follows form, as we've discussed. That we're excited to do that on behalf of on the Clario side, where we can deliver those things at throughput and scale in a cost-effective manner, different than our academic selves. So I think that what you've described is our dream, and I think that the idea to both supplement what's already out there to enhance the breadth and efficacy profiles, but then also to get ahead of new things that are coming, to incorporate this as an additional variable to consider in that multivariate analysis of how you assess efficacy. I think there's also, we talk a lot in sort of our Clario leadership group about sort of the dreams and the potentials. I mean, we talk about minimal residual disease measurement. We talk about antibody drug conjugates. This is beyond vaccines. It's in a lot of different places. There may be drugs that have been discontinued or that could be what's the word I'm thinking of, reused because they just weren't screened in the right population to begin with, right? And maybe the more we know about that, the more we can identify the appropriate target for the drug. Repurpose, that's the word I was looking for, right? So repurposing things that because of the way clinical trials have been designed in the past, maybe the distribution of people that were involved in those studies, now that we're going to know on our with our academic hats as a public resource, the variation across the world at a pretty tremendous scale, maybe we can use that data and that information to go back to things that maybe had promise, but maybe, more for specific groups than others. I mean, I think, we don't think that things just, it's not about not working. It's about how to make it as efficacious as possible and as equitable as possible.
Amazing. Yeah, what you've described in terms of, well, I think, therapeutics at the, the complete scale or gauntlet available to humanity is something that I long thought about in the world of antibiotic drug resistance, right? If you had like a globalized coordinated plan of, these antibiotics in this part of the world at this time, and then these ones over, you could constantly, you would never get these superbugs because you'd be keeping
it's like a crop rotation.
Exactly right. That's exactly right. Yeah, that's clever. Well, so it sounds to me like there's a huge data call it data lake, data refinery aspect of what you're doing, right? So that you can take a snapshot of an individual in time, but as our ability to measure health and whatever that even means, like health, wellness, diagnostics, whatever, as that changes, you actually get a very comprehensive view of an individual's natural history in their lifetime. And so they're actually a community of selves, and like you might compare yourself where you are at one point in time in your life to be more like somebody else, which I mentioned Stephen Jay Gould at the beginning of our call. I think he had I think it was stomach cancer that eventually got him, ironically probably from the asbestos in the Harvard basements, but that was never confirmed. But he was diagnosed, and he was given like an average of let's say a year or two to live, and I think he went on to live for an extra 20 years or more.
Right.
And he actually he writes about it in one of his papers where what was it? It was something to do with the median. The messages, the median, maybe that to me sounds like a My Heritage commercial in Canada. I don't think that's quite it. But anyway, the point is knowing how to compare yourself, not in the mean, that is the average, but the median segment of the population, like where is the most abundant people like me. And so what he learned is like, well, the average people who have this kind of cancer, by the way, includes people who are 70.
Right.
And so once he, being privileged with his academic background and pursuits, he was able to do a research project on his own, his own health condition, and work with his team to devise the best possible treatment plan going forward. And he would not be able to do that without the context. Otherwise, it would be average, right? And so it sounds to me like this is a future that you're building. You're on the front lines in the wet lab. We're going to need also the dry lab to complement this. So this is where I'm going with the data lake and the bioinformatics and I think probably very soon, like I'm talking years, not decades, we could be at a point where the standard of care gets better just because there's a single source of truth for all of your individual health records and biology. So that's kind of where you're going.
No, I would think that you actually bring up a point. It's a pain point in the field, actually, regardless of sort of what angle you're coming from, and something we've seen dramatically with these new methods and any kind of immunogenomics methods actually is that, like almost every other genetic or genomic field of research for the last, 50, 60, 75 years, the vast majority of research has been done in the Western world with Caucasian populations. I'm one of them. So, but they haven't really considered sex differences. Right. So in the autoimmunity case, 80% of autoimmunity cases are in women. A lot of those are antibody driven. So what is that about? And then all of the other populations around the world right, that are not really represented. And that's genomics in general. And we're working towards that, right? The Human Pangenome Project and other projects that are trying to really bring that together. When I was in Thailand recently, there's a Genomics Thailand project there that's generating tens of thousands of whole genomes, which is really exciting. But what happens, right, is where does that data go and who can access it? So these databases, and then you get into the complexity that I described earlier with the antibodies and the T cells. Not only do we need the reference to like map to, right, as a comparison point, but we also need all of those genes annotated and curated and the different alleles identified. The field needs to use consistent methods across groups to do that. And then everyone needs to be able to sort of touch the same database in as much as possible, right? Everyone doesn't release their data publicly, but like our big thousands of people population study, that's an NIH funded resource grant. And so that will all be put into the public arena for anyone to use. And so we are very dedicated on both sides of the academic and commercial venture to really broaden the resources for data analysis, because you can't see something that's not in the database, right? Because we always do these analyses in a relative manner. And so you need to have, if you're trying to map, sort of think about what we talked about with B cell development, if you're trying to map which mutations were acquired during that maturation process to change from low affinity to a high affinity, if you don't know what that original gene was, then you may completely call the mutation rate wrong. You may say, oh my gosh, there's 30% mutation, but really the original DNA allele carries 15% of that mutation. And then you're interpreting the biology wrong, right? And so I think that there's a lot to be said for just the investment and growth of resources in the immunogenomics space, that just doing this population surveillance, this isn't just, stamp collecting per se. Like, we really need to understand what the extent of the diversity is. I wouldn't say that 10,000 people is going to cover it. I would say it's a really great start, but I have no idea what it's going to take before we asymptote on diversity. We, without fail, discover something new in every individual we sequence, whether that's a new allele, a new gene, a new structural duplication or deletion. It's really incredible. I think it's rare in science to have sort of cracked open a field that people haven't explored before. It feels very, you feel very much like, and like you're going to space, except you're going to this region of the genome, but nothing is known. So everything that we identify is interesting and real. And the data component is critically important. I would say in the context of, Clario, like I was talking about before, the data that we're going to acquire is probably the most valuable asset for the company, just because it's going to be the most comprehensive data set out there. So, but these databases really, there's several of them out there. Different people use different ones. That's another issue, right? When you have a competing interest in that case. But yeah, it is desperate need for this field and many, many others.
Yeah, amazing. Just one final question on the database that you're building. So that includes the immunogenomics aspect, but I'm assuming, does that also include, like, are you integrating like electronic health records or just the natural history, or is that kind of on the roadmap to do, or what does that look like?
Yeah. So we have, basic metrics that we try to include with every data set. So sex, general geographic location, ancestral heritage, age. I think there's a lot to be done, and way more than we could talk about now about aging and immunity. But of course, as we build specific studies, either academically or commercially, we're very open to partnerships with specific topic foci. Yes, we would want to integrate with clinical records to try to draw as many correlations and associations as we can. We work with Dr. Guryari. He's our CTO, and he's an expert in developing predictive AI ML algorithms. So trying to see if we can use immune repertoire signatures as biomarkers for phenotypes of interest. There's so many places you could see where that could be useful. And again, building that database with, again, Dr. Corey Watson and Dr. William Lees, who are both very, very prominent members in the adaptive immune receptor repertoire AIR community. So we're really excited about sort of where we can take the data, and the more metadata we can get, the more information we can get, the more powerful the data becomes. It's very much on a case by case basis. But yeah, we're hungry for data. That's for sure.
Amazing. Well, listen, I mean, this could go on for hours, hours, and I think we will certainly have a follow-up. But I wanted to bring our conversation back to the preamble, which there were 3 questions I started with, and the first one was, well, what would you want to say to people if this was your last podcast, and now so you said it, but like, how would you summarize it? What's the key message here that we want to deliver people?
Right. I mean, the key message for me is that genomic variation matters, particularly immunogenomic variation matters for health, resilience, and disease, and equity and representation matter. We can't just assume that we're all the same, even within the same ancestral group. And I think it's really important that those of us that are trying to push the envelope and leave the field really try to be as inclusive and representative across as many populations as possible, because you don't want to go back and have to reinvent the wheel later, and we don't want to leave anyone out. Everyone deserves quality healthcare, and this is how we hope we're going to get there.
And so the second question was, what one person would you want your message to reach above all else? Who are you thinking of?
Right. I think as we discussed, I think that there. So I would love sort of two people to reach, I guess. I'm gonna split my gains here. So the first would be trainees, to know that no route is linear. That you learn from every experience you have, not only to take what you did learn, but also learn when to get out. Right? There are opportunities around every corner. Networks are really important. And I think mine is a great example of how you can pinball a little bit until you find a really good groove. And that's okay. I'm in my mid 40s and I'm just feeling like I'm finding my rhythm, and I'm so excited to be there. So I think it's just it's a good reminder to keep going. And I think knowing when to get out is critical, and it's not something we talk about a lot. I think the second would again be coming back to sort of the potential of the work that we're doing. I would love my message to reach people who are thinking about drug development, immunotherapy development, vaccine development, and think about how important it is to understand what the host brings to the table in terms of responsiveness, and come work with us. Come work with us at Clario so that we can demonstrate how important this is. We know it's important. We've seen it across the board. I think a lot of drug developers and whatnot get a little scared when you start talking about different population groups, right? Because a drug is so expensive to take into clinical trials and into the clinic, that the idea that there would have to be variants or a mix or a valent, multivalent approach is really intimidating. And I think the dollars just stack up in their head. But I think if it means that your therapy is more effective for more people, that should be the baseline, most important thing. And I think that we can help get a lot of people there across a variety of fields. And so I hope they listen. And I hope they recognize that getting ahead of this curve, instead of being drugged to it, once the demonstrations are there, would be really advantageous from both a, ethical and business perspectives.
Awesome. And then the third question was, what one person would you nominate to be on the DNA of things?
Yeah, I mentioned him earlier. So my mentor at Mount Sinai, Dr. Robert Sebra, Bobby Sebra, he's just an incredible personality. He's one of the smartest people I know. He's done a lot of work recently on a lot of single cell sequencing technologies and algorithm development that's incredibly fascinating. I think he could give a lot of insight into both the technology, but also the incredible potential of where that technology goes. And if you want to, I'd be happy to put in a good word.
I would love that. Yeah. That would be very helpful. It's the more we get this, we talk about knowledge translation at the beginning, right? Yeah. So, the more people that we have as part of this great conversation. I mean, just the fact that, I can say mRNA and probably at least 4 billion people now know what or have at least heard that phrase mRNA. That wasn't true 3 years ago, 4 years ago. So, the more people are part.
True.
Yeah. Yeah. So, and just, lastly, Dr. Smith, where do people find you if they want to get in touch? It's the best way.
Absolutely. So the best way is email. So feel free. My email is melissa.smith@clariobio.com.
Awesome. And you do have a lab webpage as well.
Yes. Yes, we do have a lab webpage. So you could also reach me on the academic side. So that email is ml.smith@louisville.edu. And Smith Lab at Louisville is where we are. And I would love to talk to anyone. Obviously, I'm happy to talk at any time.
Wonderful. It's been an absolute pleasure speaking with you today. Dr. Melissa Smith, thank you so much for your time and joining us on the DNA of Things here today.
Oh, it's so my pleasure. I hope we get a chance to do this again.
Absolutely.
All right. Thanks so much.
Okay, that is a wrap. Thank you so much for joining. In this episode, we traveled through the incredible landscape of immunogenomics with Dr. Melissa Smith, discovering how genetic variation shapes our immune response and the promise of precision medicine. Now, Dr. Smith's journey reminds us that science is a dynamic and evolving field, driven by curiosity and the pursuit of knowledge, truth. I hope you gained valuable insights into the importance of genetic diversity in health, not just your health, but health of humans, of humanity, our entire species, of all species for sure. And the potential understanding this diversity presents for personalized treatments. And of course, the need for inclusive and representative research to truly get a picture of the human metagenome. Our species is diverse, and it's an exciting time because we are, for the first time in human history, at a point where we're able to understand this and really quantify. So if you found this episode enlightening, please share it with your friends and colleagues. Together, we can spread the love for science, the pursuit of truth and knowledge, and its transformative power. So stay tuned for more inspiring stories and more inspiring people on the DNA of things.